Here is the lesson on cell communication. Starting with the important concepts from the last unit, which is, for the first one, it is phosphorylation. Phosphorylation is essentially adding a phosphate group to a molecule. For example, that is an ADP with another phosphate group, and that turns into ATP. That is one process. Another concept to understand from the last unit are enzymes. Enzymes are essentially proteins that are catalysts which speed up a reaction by lowering the activation energy. For example, if you have a reaction and this was the rate of the reaction, that would be normal without an enzyme. However, if you add an enzyme, the activation energy lowers, which in turn makes the reaction go faster. Enzymes work by a substrate attaching to the active site of an enzyme. For example, if you have the enzyme, it has a pocket to where the substrate will attach to. That is called the active site. The substrate should be in a perfect shape to attach to the active site. This creates the substrate enzyme complex, which makes the reaction happen. Here are some better pictures of phosphorylation and enzymes. Let's get to what cell communication really is. So we'll start with the G protein pathway, which is the most common type of pathway. So let's just say I have a phospholipid bilayer, and you guys should know what it looks like, so I'm just gonna draw a bad one. We'll just say this is the phospholipid bilayer. So in the bilayer, there's something called a G protein linked receptor, and we'll just say the protein looks like this. All right. And basically that receives a signal from a messenger, which is called a ligand, and we'll just say this is a ligand. And that ligand will attach to the point where there is almost like an active site from an enzyme. It will attach in this pocket. In the pathway, there are also proteins that are called G proteins, which relay the message to the location where the message is supposed to go. After the ligand binds to the G protein linked receptor, these G proteins break off, or they dissociate. Whenever the ligand binds to the G protein linked receptor, it goes through something called a conformational change, which is essentially a shape change to where it has a different function than what it was before. This is the beginning of a signal transduction pathway, which is the series of chemical events that take place from the ligand binding to an actual response by the cell, which is like mitosis and other cell functions. So now the dissociated G proteins will interact with other enzymes, such as this one, and the enzyme will begin making like other molecules. And whenever they make other molecules, those molecules are called secondary messengers. Because the binding of one ligand results in many secondary messengers, this is called signal amplification. And one example of a secondary messenger is called cyclic AMP. Eventually, the secondary messengers will relay all the way towards a target protein, and this results in a cellular response. Here's a better picture of the G protein pathway. The next pathway we're going to talk about is the tyrosine kinase pathway. So imagine we had a cell membrane. There are two receptors, which is called the tyrosine kinase receptors. So we'll just say like that. And just like the G protein, it gets a message from the ligands. And then the ligands attach to the binding site. Whenever it attaches to the binding site, both of these tyrosine kinases go through something called dimerization, which is basically whenever these two receptors join together. On these tyrosine kinases, they have tyrosines on each. And right now, they are unphosphorylated. And both of these tyrosine kinases do something called cross-phosphorylation, which is adding a phosphate group to each one of them, and they cross-phosphorylate each other. Now that the tyrosines are phosphorylated, they can now attach to a protein. We'll say this is the protein. And this will make a cellular response. Here's a better picture of the tyrosine kinase pathway. The next pathway we're going to talk about are ion channel receptors, or also known as ligand-gated ion channels. So let's say we had cell membrane. There would be a protein right here. And this is where the ligand would bind to the site. So here's our ligand. It would bind to the site like that. And whenever it binds, it opens a gate. So this would be the inside of the cell. It would open a gate. This would allow ions from the outside to flow inside the cell. An example of this would be nerve signals, signaling to muscles to contract and stuff like that. Here's a better picture of what ion channel receptors look like. The next pathway we're going to talk about are intracellular receptors. So with the other pathways that we were just talking about, those were dealing with hydrophilic molecules. And with hydrophilic molecules, they cannot just simply diffuse through the cell membrane or the phospholipid bilayer. For intracellular receptors, they deal with hydrophobic molecules. So let's just say these are our hydrophobic molecules. And we're going to have our receptors right here. And these receptors are either in the cytoplasm or the nucleus. Basically, all these molecules will do, they'll just simply diffuse through the cell membrane, and they will bind to the receptors inside the cell. After they bind, especially in the nucleus, we'll say this receptor is in the nucleus, then it will have something called a transcription factor, which is basically just making RNA. So the responsibility for this receptor, after it receives the message, it will start making mRNA. Here's a better picture of the intracellular receptors. Now we're going to talk about our secondary messengers more specifically, and these are the relay molecules in a cell's cytoplasm. So the first one we're going to talk about is cyclic AMP, or just ZAMP for short. So we'll say again, we'll use the G protein pathway as an example. We'll draw the membrane, and we'll draw the receptor, and then we'll say these are the relay proteins, and this is the ligand that will attach. So as we said before, whenever the ligand binds to the G protein linked receptor, these relay proteins, they dissociate. And more specifically, this is called the alpha, and this is the beta, and this is the gamma. And whenever they dissociate, the alpha, it has GDP, and it exchanges it for GTP. This protein will then bind to something called adenylyl cyclase, and this basically makes the cAMP. Here's a better picture of cyclic AMP. The next secondary messenger we are going to talk about is actually Ca++ or Ca2+ and IP3. So say we had, obviously another cell membrane. There's going to be a PIP2 molecule. And basically this just diffuses straight through the membrane, and it goes through a protein which is called phospholipase C. We'll just say this is it. And this basically just cuts this PIP2 in half, and it turns into IP3 and DAG. So now we have the IP3, and we'll say this is the ER, the endoplasmic reticulum. And on the ER, there is a receptor which is called the IP3 or Ca2+ channel. And basically, this IP3 binds to the channel, and the calciums are able to leave the ER, and because they just go from high concentration to low concentration. And now the cytoplasm has higher concentration of calcium. Here's a better picture of Ca2+ and the IP3 as second messengers. Thanks for watching my lesson on cell communication.